We're going to look at humoral immunity, which is a specific immune response which centers around the fluids in the body. And the fluids are the blood system and the lymph system. And it's called humoral because in the olden days of Greek times, I believe, the humors were your fluids. And if you were in good humor, I think it was that your fluids were in good order. And in the humoral response, we are looking, because it's specific, as opposed to the non-specific, which is just any general antigen that's not self, we are looking at specific antigenic variants, which will be a specific sequence of amino acids on a protein, or a glycoprotein, or a glycolipid, or a polysaccharide of a pathogen, which is not self. And our body's able to recognize it as not self because, amazingly, when we're embryos, we edit our T and B cells to make sure that any that have receptors which would be complementary to our self and our own peptides, etc., anything that would attack our own cells is deleted, leaving us with millions and millions and millions of versions of T and B cell receptors which are capable of binding to pathogens which you either have met or haven't met or may never meet. They're circulating in your body now. All of these T and B cell receptors for variations of polypeptides, for example. And so the humoral system is actually going to end up with us forming antibodies. And the reason why it's relevant of it being the fluids of the body is that antibodies, and we can see them at the bottom here, are soluble. And so soluble antibodies are able to get all around the body, and because they dissolve in the blood plasma, they can go through our circulatory system, they can go through the lymph system, and they effectively lead pathogens with no place to hide. And they greatly increase the speed at which we're able to destroy the pathogens because the fluids of our body are obviously moving all the time, and there's a much better chance that there's going to be a collision between antibody and the pathogen. And so how does this work? Well, with all specific immunity starts with antigen presentation. And this will be by one of these two cells. This is a dendritic cell, and this is a macrophage. And the dendritic cell and macrophages are called antigen presenting cells, which mean, and this is this diagram here, it means that what they do is they take a specific antigen, which, as I say, might be a specific sequence of amino acids on the surface of a pathogen. It wouldn't be the whole pathogen, it would just be a small sequence of non-self that the dendritic cell or macrophage recognizes. And it digests the antigen, that will actually digest the whole pathogen. In endocytosis, the whole pathogen, it forms a phagolysosome by fusing the phagosome and the lysosome. And the lysosome, in this case, is an antigen presenting cell, partially digests the pathogen and then presents on its surface with using MHC2 protein, the antigen. And MHC2 is a protein which holds the antigen on the surface of the macrophage or the dendritic cell and presents to the rest of the immune system the antigen. And what this macrophage or this dendritic cell is effectively doing is it's saying, I found something that's not self, I've digested it, here it is, here's an example of this pathogen that's inside us. Is anybody interested in this? Is anybody interested? Here it is, I'm holding it in my MHC2 protein. Now, the lymph system, which is the fluid system that runs kind of behind our circulatory system, our blood system, has a number of lymph nodes in it, which are concentrated areas where lymphocytes, white blood cells, hang out, particularly dendritic cells, macrophages, T cells, and B cells. And when a macrophage presents antigen in MHC2, when it travels around the lymph system and travels through a lymph node, it is likely to come across a T cell. And a T cell will bear a receptor. And there are millions and millions and millions of versions of T cells with millions and millions of versions of T cell receptors in your body. They're circulating right now. And the the antigen that I've drawn here will have a T cell which is complementary to, and this is interesting, not just the antigen, but the antigen being held by the MHC2 protein. So here we have the way a T cell receptor looks, and that will have a very specific set of amino acids which will be complementary to the antigen being held by the MHC2. So it recognizes both, really. It wouldn't recognize the antigen on its own. It needs to be held by MHC2. And what that does is when in the lymph node the T cell meets the macrophage or the dendritic cell holding in its MHC2 this antigen, hello, anybody interested in this? The T cell effectively says, I've got the complementary receptor. And they bond. They bind together, and you can see in this diagram here the way that happens. So this is a dendritic cell on the left here, and you can see as a result of a complex of proteins, a bit similar, we've met complex of proteins before in transcription. Remember, transcription factors come together to form a complex rather than it just being one protein. And here we have a number of proteins which cause a signaling between these two cells. And signaling is a really important word here, because once you have the binding of the T cell receptor and the MHC2 holding the antigen, you get, as a result of the binding of other proteins, a release of cytokines, which are immune chemicals. And one thing actually I do want to mention here, which is very important, is the protein CD4, which is on a T cell, a T helper cell specifically. And this CD4 helps the MHC2 holding the antigen bind to the T cell. Now, the reason I wanted to mention CD4 here is it's going to be relevant for us when we look at HIV later, because HIV attacks these T helper cells. And the reason it can attack them is it is able to attach to the CD4 that T cell helper cells express. And so there we have the T cell, the T helper cell, and the dendritic antigen presenting cell. They bound together, and there's now going to be a release of cytokines by the macrophage, which is going to stimulate this T helper cell. And the T helper cell is going to do two things. It's going to release cytokines which stimulate the antigen presenting cell to become more aggressive in its phagocytosing. And then, really interestingly, it also produces cytokines which activate itself. And part of that activation is that it releases the chemical, and it also forms receptors on the surface which would respond to that very chemical it's released. This is called an autocrine response. We've met endocrine responses with hormones which travel around the body to target cells. Paracrine is a response that's to cells around nearby, and autocrine is when something releases a chemical, a cell releases a chemical, and it has an effect on itself. And so this actually causes a massive proliferation, which means replication by mitosis, of this particular T helper cell, which bears this particular T cell receptor. Remember, this T cell receptor matches the antigen on this pathogen that we've met here. And so this is the relevant one. And so of these millions of T cells roaming around your body, this is the one. I'm interested in that antigen held in MHC. I'm the one. And the body effectively says, if you're the one, we need loads of you then. And we get this massive proliferation of this type of T cell. They are circulating in the lymph, for example, and as a result, there's a much bigger chance that they are going to come across other immune cells. I say other immune cells because now we need to meet another side of the story. We've had the dendritic cell in the macrophage presenting antigen. There's another antigen presenting cell in the humoral response, and this is the B cell here. And the B cell will have, remember, the pathogens don't go around in ones and twos. There's usually thousands of them, and they're reproducing exponentially, which is why we have to try and get hold of them and kill them. A B cell will have met the same pathogen, and B cells have on them membrane-bound antibodies. And this is an antibody here, as we mentioned. And the membrane-bound antibody, the membrane-bound antibody, there'll be lots of those on a particular B cell. And like the millions of versions of T cells, there's millions of versions of a certain antigen binding site on a T cell, which is here. Sorry, an antibody, which is here and here. And they will have many, many variations for pathogens you've met and pathogens you haven't met and pathogens you'll never meet. If it's not yourself, there's loads of variations going on in your body. And so there will be a B cell that will exactly match the pathogen, that the antigen, that the dendritic and macrophage met earlier. And this B cell will do exactly the same, because it's an antigen presenting cell, as the macrophage and the dendritic cell did. It will endocytose, it will partially digest, it will then present with MHC2 on its cell membrane. And so we have a B cell which is presenting with MHC2, the antigen. Now, in the lymph nodes, for example, the T cells, which have been massively proliferated, on one of them is now bearing the complementary receptor for that antigen in MHC2, will then dock. And here we have an example of this here. This is a much better diagram than I can do, obviously. We have the B cell in red and the T helper cell in brown. And you can see the T cell receptor and the B cell, they link, and we've got other proteins there which are formed in the binding. And in fact, you can actually just see there that there is CD4, the green thing, once again, which helps a T cell link into MHC2 carrying antigen. And as before, I can get rid of some of this stuff now. As before, when the T cell met the dendritic cell or the macrophage, we get an activation. We get an activation of the B cell due to cytokines which enter into the B cell. And that causes a couple of things to happen. The first thing that is going to happen is that we are going to activate this B cell to proliferate, because it is one of the chosen ones. You are the relevant B cell with the right antibody on that matches which is complementary to our antigen. So we need loads of you. And then those B cells, a number of them, are going to turn into what we call effector cells. And effector cells are cells that are going to have a real effect on this, and they're called plasma cells. And plasma cells take this humoral response to the new level, which infiltrates all fluids of the body. Because if we look at this B cell that we've looked at here, in order for the membrane-bound antibodies that are on the B cell to interact with antigen, that you're only as effective as the whole B cell interacting with the pathogen. And that is going to happen over a long period of time, I imagine. But wouldn't it be wonderful if you could, in fact, free the individual antibodies so they could flood all the fluids of your body and therefore allow them to independently go out and bind to pathogens? And that's exactly what happens. Plasma cells here produce, through massive amounts of protein synthesis, so you can imagine that their ribosomes and their rough endoplasmic reticulum and their Golgi apparatus are going nuts in there as a result of the cell signals of the activation of the T cell, produce free standing antibodies like this, which then flood the humoral fluids of the body, the blood, the lymph. And this leaves, as I mentioned earlier, the pathogen nowhere to hide at all. And it greatly increases the speed and the likelihood of the collision of complementary antibody and antigen. And when the antibodies bind to the pathogen, they're going to have a number of effects which lead to the destruction. Now, antibodies do not destroy pathogens by themselves. They effectively aid the process. And now we're just going to quickly look at how that happens. Before we do that, I just would like to mention that as well as the proliferation of B cells, T cells also stimulate the production of B memory cells and T memory cells, which are going to be very relevant to the bottom right hand corner here, guys, which is immunological memory. And that's very relevant to vaccination, and we're going to look at that later. But that leaves in the organism and in you an ability to react faster and with greater volume of antibodies in the future if you meet the same disease again. But let's go back to how antibodies actually lead to the destruction of pathogens. We have here a diagram which gives an amazingly detailed version of how antibodies are going to lead to the destruction of those pathogens. They are all of interest to us as biologists. For A-level, though, there's a few that I really want to point out to you. So the first is we'll start with neutralization on the left. And this is a really important one, because what it does is it stops the pathogen from carrying out its normal cellular activities. And the way that we now know that cells and cells interact is as a result of the binding of receptors, for example, or recognition proteins or glycoproteins on the cell surface membrane. And if antibodies come along and bind to the membranes of the pathogens, then they block the binding sites that will be used for the pathogen. And so you can imagine if a virus is going to land on a cell membrane and it's going to dock in with the receptors, if you cover those receptors, then it can't dock. And so therefore you neutralize the pathogen because you block its binding sites or its receptors. And so we call that neutralization, and that's a really important one. So that's number one. The second one is agglutination. And this is very clever. If you think about phagocytosis, if it happened one by one, and you're talking about millions of bacteria, then a phagocyte is going to do one, engulf, digest, two. But wouldn't it be fantastic, instead of doing them individually and coming across them on a one-by-one basis, if they could all be clumped together for phagocytosis? And this is what agglutination does. Quite incredible. Because, I'm going to just do a little drawing here. Because antibodies are Y-shaped molecules, they have 1, 2 antigen binding sites, which means that an antibody is able to bind 2 different antigens and therefore 2 different pathogens. Antibodies have got a hinge here, okay, and they can actually go so they are pretty much at 90 degrees to each other. And so you'd have pathogen there. And this means that you are able to group together, and you can see that agglutination is a bit like glue together pathogens by the fact that antibodies can bind 2 at a time. And if you wanted an analogy of a modern thing, if you've ever seen those adverts where it says, take all your individual loans and consolidate them into one easy to pay big loan, it's a bit like that. It takes all of the bacteria and it pulls them together into a clump. And so when a phagocyte comes along, they are all in one area, and it's able to phagocytose and digest those pathogens much more quickly and much more efficiently. Okay, so now number 3 and 4 are not specifically ones that we need to know that much about. 3 would be relevant for those soluble antigens, which might be small proteins or toxins, and antibodies, because onto them, are able to cause them to come out of solution, and we call that precipitation. You may remember in chemistry you have precipitation reactions, which form a solid. Complement system is a really clever system which causes foreign cells to have holes punched in them, which leads to cell lysis, you can see here. But the one that I want to focus in, as well as neutralization and agglutination, is the enhancing phagocytosis, specifically in a process called opsonization. And opsonization is a process by which, if we did have a situation where a phagocyte came across a group of pathogens, for example, like this, that were bound by antibody, it would it might, engulf them in a general way, and that might be fine. There's our macrophage or whatever. But it would be wonderful, wouldn't it, if there was some kind of docking on the antibody, which, a bit like a guy at an airport with the lights bringing a plane in, is able to cause a complementary binding of the antibody, which is bound to the pathogen, into a receptor on the phagocyte. And lo and behold, this is exactly what happens. So if you have an antibody and you take the constant region here, remember there's constant and variable regions. The constant region here is going to be complementary to a receptor on a phagocyte, a macrophage, for example. And so that means that there is a docking of this antibody into the macrophage, and it's called opsonization because it makes the whole interaction between the phagocyte and the antibody, which is binding to the pathogens, much more efficient and smooth. And it sounds, opsonization sounds a bit like optimal, and it is a bit like that. It makes it a much better phagocytosis experience, faster and more efficient, because there is a docking mechanism which leads to the areas where the phagocyte can link into the antibody. And so just going over those three again, you can see the ones that were relevant to us. You can see that the antibodies don't cause the destruction. They either leave them with neutralization as sitting ducks waiting for phagocytes, agglutination, which groups them together, waiting for phagocytes in one easy to manage clump, or they opsonize, which is they create docking stations for the phagocytes, which make phagocytosis much more efficient. Okay, so that is the ways that antibodies work. And if we go back to our diagram, we can see that the antibody here is the final, at the bottom, is the final part of the humoral response, which enables us to flood the lymph and the blood with those antibodies, which do the neutralization, agglutination, and opsonization. And so the humoral pathway has been antigen presentation, T-cell activation, B-cell activation, the production of effector cells, called plasma cells, the production of soluble antibodies, which then flood the system. On the back of that, we also have the production of B memory cells, which lead to immunological memory. So that is the humoral response in the specific immune system. The other side of it is the cytotoxic or cellular immune system, which deals with cells that have been infected by viruses, which I deal with on another recording.